Hi, everybody. Welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is Thursday, August 25th, 2022. We're back from summer break. Today we welcome Andrew Maurer, who is assistant professor in the Department of Neuroscience at the University of Florida. His work has visited a wide range of topics, including dynamics of population activity in dopaminergic neurons, the effects of ketogenic diet on the brain, vagus stimulation effects on learning, and the function of the hippocampus and related structures in memory and some other things. One thread that connects all, or at least most of these things, is the rejection of this sort of linear feed-forward model of neural connections and embracing the reentrant, recurrent structure of brain circuits that drives oscillations and more complicated patterns like turbulence. In the hippocampus, reentrant collective activity of cell populations is often assessed from oscillatory field potentials. And I think this is largely what we'll be talking about today, but who knows? So welcome, Drew.
Welcome. Thanks to be here.
Also with us today is Francesco Savelli, UTSA's own expert on hippocampus dynamics.
Hello.
Hi, Francesco.
Hi.
And James Jones, a PhD student in the neuroscience program at UTSA. Hi, James.
Hi.
And I'm Charlie Wilson. So, Drew, regular listeners to the audio podcast have most recently heard a retrospective on a podcast we recorded with the late, great John Lissman back in 2008. At that time, John was working on his now classic work on the theta-gamma coding idea in the hippocampus. The key part of this was the idea that the meaning of an action potential in a hippocampal neuron was embedded in its relative timing to the ongoing theta oscillation rather than just to its rate. And he thought neurons on the receiving end of those hippocampal neurons ought to have access to their own copy of the theta oscillation, perhaps at phases different from the one at the origin, so that they could use it to decode the phase information on their inputs. In our podcast, John argued the case for studying coding schemes of this sort as opposed to rate codes. So this was 14 years ago, and I'm sure that a lot of things have changed. So Drew and Francesco, could the two of you start by bringing us up to date on theta oscillations, theta phase, and the language of the hippocampus neuron as we know it now? Is that a fair question?
It is, yeah. But I will defer first to Andy.
This is a lot to unpack.
Yeah, it is.
I think it was for the idea at the time was avant-garde, right? It was people were very much working to reconstruct the rat's position based on the knowledge of when the neuron fired an action potential. And they found out that they could improve that a little bit by using the phase information. And I think John's idea was very much along these lines, in which you could have a rate code, but you could improve that code if you include the phase in which the neuron fired relative to theta.
Yeah.
And he discussed multiple coding schemes. I think somebody asked him a question about how many coding schemes are there, and he showed his comedic deference in replying to that answer. And so I'll ask Francesco, how many coding schemes are there?
Oh, I think I don't know. I think probably you can find many. I think the holy grail is to find one that has, I would say, I would try to say purposeful readout, right? Because there's a lot going on in the brain, and it correlates with internal or external variables. So, to really know which scheme the brain is using, somehow you gotta catch the brain using it.
Yeah.
And I think that's still what is missing. And I don't know if I'm anthropomorphizing in a way, just because, you always think about okay, you're always trying to put the homunculus back into the brain, saying purposeful readout. But I don't know that I have a better way to think about this. How do you know what the neural coding scheme that matters is if you don't know what matters to the brain, at least in the context of some function? So I really think, I'm really definitely interested in all the temporal strategies of encoding information, I think it's really important. I consider, for example, phase precession of place cells like one of the, probably the most important observation in the physiology of the hippocampus after the observation of place cells.
So John interpreted phase precession in a kind of funny way, I thought. I mean, it didn't sound that familiar to me. He was saying that the phase told you about how far into the future the hippocampus cell is predicting. So where does that idea come from? And is that still an idea about phase precession, that it has to do with time, not place, or time and place?
The idea of it anticipating the future is that if you had three cells that were overlapping, A, B, and C, and they were overlapping, so the rat would run into place field A first, then B and C, but they overlap spatially. Within each theta cycle, it would fire A, B, C, and the next cycle A, B, C. And so what you would see is if you took your temporal window and looked at really small, you would see these little sequences of activity that repeat each theta cycle. And it was as if the rat was replaying or anticipating the future trajectory by providing this little sweep forward. Looking to C, looking to forward to C, and then eventually A would stop firing, and then you'd have place field D pick up. And so the rats begin, the trajectory, the sequence of activity begins to anticipate place field D. And I think that idea is pretty much, I would wager that most card-carrying neurophysiologists would agree with that description today.
Yeah, I think it's there. The issue is, I don't know if I'm characterizing this problem correctly, but I think the issue is also, do you get that sequences as a byproduct of the place cells engaging in phase precession? So because there is phase precession, then automatically you get, as a fallout, you get those sequences in the phase, or you get phase precession because there is this kind of like sequence action of the cells propagating, the activity propagating into these routes in the network, paths in the network. And so I don't know that we have an answer to that. I mean, it's like, but I think, again, when I say that is the phase precession, I consider it in a way the most important observation. I mean, it's really telling us something, which we don't know what it is, but somehow you know that it's telling us something. And, again, it goes back to, is it epiphenomenal or is it purposeful? And whatever that means, but that's the way I think about it. It's like, and so, it might be epiphenomenal, but then it's telling us something about the dynamics of the circuit. Or it could be like something that is really shaping up some packets of information in some way that is relevant to cognition, for example, of some sort.
So if the theta is putting a timestamp on spikes, and that's part of the code, then doesn't the neuron that's receiving that information also need to have a copy of that clock to know what the phase is of its incoming synapses? So John said, yeah, maybe all the different targets have theta oscillation at different phases, and they read out the neuron that's telling the future, the present, or the past.
I think that Francesco is touching on, and that's almost what's missing. That's a direct hypothesis, is we should eventually find these neurons that are sensitive to that afferent input, and I don't think we have yet.
No, so there is this study by Tingley and Buzagi's lab. So they looked at the relationship between hippocampus and the lateral septum. If I'm correct, it's the lateral septum, right? And so they found cells in the lateral septum that they have the phase precession. So they have that kind of the temporal code, the phase code. But as the rat was running around, you didn't have a lot of graded firing rate differences. And so, one explanation of that was exactly what Lisman was saying, which is, see, when it's communicating to another structure, the firing rate modulation does not matter. What matters is this temporal code. there was still rate modulation in the distributions of the plots, and so you always wonder, like, the lateral septum is kind of a smaller structure, gets input from all hippocampus. Is it possible that these was, place cells that have such a big place fields that incorporated the entire apparatus, right? Because when you look at the distribution, there was some rate modulation. But I think what is really missing, the most important question is, you can think about, there is a lot that has been done about this phase code and the ability of relating to sequences. And so the idea is that, is the hippocampus about sequences? And so, like, if you have a rat running on a track, the succession of place cells spatially is just one instantiation of this ability of the hippocampus to create sequences. It's nothing special about space. Or is it the other way around? The old the cognitive map theory. The spatial framework is a fundamental constituent ingredient of the memory. And I think I was talking to you the other day, if we, this can only be resolved if we look at the animal going in a two-dimensional environment, right? Because now, what is phase precession in two dimension? There are some studies, very few studies that have analyzed it, but the place fields are small, so the animal is kind of traversing. I think it's still like we don't have a very good grasp of what phase precession or what this sequence encoding would be in two dimensions. And the reason I insist on that is that, yes, you might say the firing rate does not matter, but it exists. The place cell fires when the animal goes back there, no matter what path the animal has taken. So there is path equivalence. That is what is, in essence, the allocentric property of this firing, which is what inspires the idea of the cognitive map. And so okay, I'm perfectly happy to try to reconcile sequences with the cognitive map, but the fact is, when it's firing, if you get a grid cell, as people say, okay, that's not what the brain sees when a grid cell fires, because that's an average that the experimenter has done. But it is true that those locations are pretty stable, so when the animal goes back, no matter what the path. So there is an allocentric definition. So now the question is, when it comes to phase precession, is phase precession disease, are they allocentric or not? Nobody has looked at it. I don't even know how exactly to frame that question, but it's like, it's true that you can add positional information through phase precession. Can you do it also in 2D, and how well? And if not, well, if it is, well, then we know that we can reconcile cognitive map with this sequence and temporal code. If it's not, then it means that the rate is telling you something more than the temporal code.
You would be breaking the Jensen and Listman reconstruction paper by showing that phase. Yeah, that's true. So what Francesco is saying is we very much get fixated on our favorite experimental approaches. And when we have rats running linear tracks, it's easy to have place cells fire in sequences. But now if we're in two dimensions and my rat's foraging for chocolate sprinkles in the environment, and I have one place field here and another place field here, my rat can run this way, and it can run this way. And so in one instance, A fires before B, and the other is B fires before A. The other thing that's happening that's interesting is the neuron always fires at a latent phase as it enters the field. Now, if it enters the field, it would fire early here. If it entered the field here, it would fire early here. Then the phase gets dissociative spatial position. So then you would break you would actually have an instance where you would include a new variable, the phase in which the neuron fired, and but you would increase your uncertainty about where may not improve your certainty about where the end
right.
was.
So yeah, exactly. So does this argument that the temporal code adds, improves the estimate, does it extend to two dimension? Or if not, well, then we have found a case in which the rate code tells you something more than the temporal code, because it's still, that cell knows where the animal is. I don't know whether the animal knows where it is because that cell is firing, but I know that cell knows, so to speak, of course.
So that it seems to me that if we took the phase code seriously, we would make a heat map of phase instead of a heat map of rate. So when we if people make a place cell diagram, they show some heat map of how fast the neuron was firing depending on where the rat was. I've never seen one where they show the phase on theta as a function of where the rat is.
I'll send you a picture. It was a Skaggs 1996. He color-coded phase.
Yeah,
And
okay.
he made like little, it was, so he did descending phase and rising phase, and he colored them as two colors. And there was an annulus around the center of the, so there's a big red on the outside and a blue in the center.
That was still a maze, right?
No,
There-
it
it was 2D?
It was 2D.
Okay.
But and so when Francesco brings that up, I start to think like, yeah, no, and you said another interesting thing. You said homunculus because a code always implies a reader, right?
Yeah.
So who's reading the code? We make it I always think when I use the word code, I mean that for me, but I don't know if the brain's using that code.
Yeah. Yeah, and so like I'm aware that I'm at risk of, just falling into that. Okay, I'm trying to put an homunculus back and expecting that, but at the same time, I don't know how to break out of that way of thinking because in the end, if I'm trying to, and then it goes back to a bigger question, right? It's like, why is it that everything that happens in the brain and neuron firing, it has to have a representation of something?
Who reads the reader? Who encodes the encoder? Who trains these representations?
I'm wary of pushing the computational interpretation too much. But let's just say, you're writing a program, right? Doing something, data analysis, whatever you want. Are all the variables that you use about representing the salient information? How many variables you use just to do auxiliary operations? And so it's like, that's always like when they say, thalamus has these many cells and cortex has so many more cells or whatever. It's like, but this is for information. So, okay, maybe there is redundancy for error correction or whatever. But maybe cortex is doing something and needs variables to do that. I know I'm pushing the computational idea too much. And there is always this question about is really algorithms and data separated in the brain, or is it more like, it's something together? I think we talked about in another podcast about this, so I'm not going to repeat myself, but does it really have to? In neuron firing, is it computation or is it data? It's like, is it an algorithm or is it data? It's like, so it goes back to that. So are we trying to, if we're trying always to think about everything a neuron does is some kind of neural coding of what? It's like, it's just, so.
Yes, I think, one of the things that strikes me about almost everything that could be called neural correlates of behavior kind of research is that somebody's recording a neuron in some kind of consequential situation for an animal, and then they say, well, this neuron has in its firing some information about this and about this and about this and about this and about this. And so you can do that. You can pull that out and you can show, yes, if as an observer, I can know this about the animal and that about the world and that and that. But the question is whether those things that neuron is embedding in its firing are actually used by anybody to do anything.
Yeah.
So it seems almost necessary to look at the next place. But of course, one of the things Drew points out is there isn't really a next place. Everything is kind of the same place.
I always thought that everybody loved the homunculus argument so much that the homunculus ended up being the prefrontal cortex or the hippocampus. You just keep rallying it up the chain until you find the executive, and somehow it just figures things out. But yeah, with a loop, it ends up being a very distributed process. It's in that way, things get interesting when you were saying, is it the algorithm, or can you expand on that? The algorithm or the data?
Yeah. So, and this is really goes even in cognitive science. A lot of neurophysiologists don't. There seems to be this rivalry between cognitive scientists and neurophysiologists, which I don't necessarily buy into. But it's the idea of the cognitive science is like all the cognitive operation can have a computational representation. So it's like there's a computational process and a representation. So in a way, in computer science terms, it's like, okay, you got an algorithm and you got data that work you got an algorithm that works on data. And so the question is, in the brain, there's neuron firing, and where is the data? What is the algorithm? What is the computational process that acts on, some kind of representation? Are they really that separated, or is it like more everything together? Yeah, that's.
So I'd like to know how you get them to be both in the same thing. I think of, if I'm a neuron, my inputs are the data,
Okay.
and the algorithm is whatever I use to make my firing pattern based on those inputs.
Okay.
Is there a way as a neuron that I could be the algorithm and the data at the same time, other than just by...
Well, basically, you're saying, okay, so that's one view, which is perfectly, acceptable. I'm sure it happens probably in the brain somewhere, but it's like you get your data as your spikes, and you see the neuron as a computational unit that then transforms that into something else, and there is an output. But, people also study about the input of a network, right? So people talk about computational operations of a network. So people just like, okay, can you do a division? Can you go, all that kind of stuff? And people used to say, okay, you can make it in a dendrite versus you can make it in a network. So now if you're talking about a network so now those neurons, what role do they have?
Yeah.
Okay, so and
because the network is creating data for itself.
Yeah,
basically.
So it's just hard to separate what we call an algorithm from data. So, but is this even relevant? Is it just an academic point or
philosophical?
A philosophical point? But it is, I think, in my opinion, relevant to the extent that we have this tendency of, looking at, what a neuron does and trying to, think that that's just coding for something. That activity is coding for something, and so. That's not everybody, the recent work by Buzaghi, for example, it's just like, okay, no, we don't want to look at things that way. But there is still these kind of things.
We were influenced by communication technology. Like right from the very beginning, Sherrington was impressed with the telephone network and the
World War II.
And we've just been more and more interested in communication technology and thinking of the brain as a communication machine. Mm-hmm. And I just can't think of an alternative way to think about it other than...
Well, so I think you have to start stripping out the codes in the encoder and the decoder. I've been toying around with this idea of... I was taught in school to never use the definition, use the word in its own definition, right? But what I started thinking about what the brain does is almost being circular. It's almost self-referential. The visual cortex eventually projects to the hippocampus, and the hippocampus projects out the visual cortex. And by taking these, it knows it got that stimulus because the hippocampal neuron fired. The hippocampal neuron helps the visual cortex know it's correct by sending a feedback. And so they're almost pointing at each other like, I know I'm right because you're right. And so then what you end up with is a distributed system where...
No wonder we're wrong so much.
Yeah, because we're all wrong, so we're all pointing away from each other.
So would you guys suggest that we move towards trying to record network activity? I mean, people have been recording neuron activity, and it sounds like both of you are considering the network and the importance of the network, and that the neuron might just be one facet or projection of the network onto some way that we can visualize it easily. I guess if we're going to move towards the network, what are there approaches right now in the hippocampal field that are being used? I know neuropixels is a new technique that's come out that allows for recording of multiple neurons.
It's still neurons, right?
Yeah.
So is there such a thing as a network beyond the neurons that you could measure and query about things, or is it really just neurons? One of the things that John Lissman said in the podcast that we are referring to a lot was, when you take a look at the brain, open it up and look in there, there's just a bunch of what's in there. It's just a bunch of cells, right? That's why he started out by saying that. And to some extent, that's true. The network is something it's a kind of a construct that we made up. What we can see is in there is some cells.
Connected.
Yeah, so tell me why.
Connected.
Yeah.
Where there's way more synapses than there are neurons.
Yeah. But still, there's nothing to measure except the neurons. There's not some magical emergent something there that the neurons don't make. No, like hovering aura or.
Yeah, I guess to like rephrase my question.
Well, you measure neurons.
Yeah.
Yeah.
Not neurons.
Also, your issue is just that everybody has to measure 2 neurons and not 1 neuron.
No, well, the question is like, there is a lot that, the issue is like, is there multiple neurons connected to each other? What do they do? Because why are they connected to each other? What do they do connected to each other? It's so much that a neuron, I mean, even just like you can, depending on at what level you unpack your universe, right? You can take a neuron, I call it, and you could think of it perhaps as a network of ion channels. Yeah. Right, you think about neurons, like, and it's just good. There's a universe there, right? So the issue is like always, okay, if you have a lot of neurons, what is that they are? Are they cooperating in somehow?
Yeah.
Why is it if I look at different place cells, if I look at 3 place cell in one environment, and I can look only at what 2 are doing in another environment, that doesn't tell me what the third one does. But in grid cells, I can predict what the third one does because, they remain coherent. What does that mean? Why does it do it that way in one way? Why does it do it the way or another way? So there's like some operation that the network does. And so that's just like you need to study those things in a way to just kind of observe as many neurons as possible at the same time. But I agree, you're still observing what the single neurons do, and you're just putting everything together.
Conceptually, the idea that neurons act as a group and do things in a network isn't new at all. I mean, it's part of sharing. It's not like we've made this huge conceptual breakthrough and we suddenly realize now that neurons are connected and they work together as a group. What's new is just the ability to record the activity of lots of neurons at once.
Right.
And so I would not call that a conceptual breakthrough. I would call that maybe a technical breakthrough.
A technical breakthrough. Yeah, no, I mean, that is a technical breakthrough, but then there are all new things that you might learn about how the brain works when you look at many neurons at the same time. The same way, you just learn so many more things about a neuron when you can do, intracellular recording versus extracellular recordings, right? So it's just like you can observe more and hopefully something. But your question was like, more about methods, concepts.
I'll apply the question. Like, are there any cool new results about networks of hippocampal neurons? Like, people have recorded single neurons and they fire preferentially at a certain place. And that's a very cool finding, and it's been repeated like a ton of times. Are there findings now that show like interesting dynamics between those neurons and their respective place fields? Like, I guess what does the field look like right now in terms of like network recordings and results from those recordings?
So I mentioned, I think, for example, one striking thing is this a bit, just grid cells seems to be very concerted activity. Mm-hmm. So that the relationships of the firing rate maps, okay, of 2 grid cells, mm-hmm. Remain constant, and if with some caveats, but they remain constant if they're grid cells so-called in the same so-called module in different environments. So it's like a rigid system to some extent. It's a constrained system. Place cells don't do that, don't have that, or at least I would say is like most of the consensus that they don't have. As a matter of fact, they have this ability of reshaping the whole code. There is some data indicated at a very local level that might not be true, but in general, it's just such a big difference, right? So, what is it telling us? and again, we don't know, it's like phase precession, right? But it's telling us something about how the circuit is organized and potentially something about functional, the function of all this. So
if we carried this right to its ultimate extreme, we would see all the neurons all the time. And then we could do the same old experiment we always did, have a rat run on a track or have a rat forage in a space, and now we would see the entire network doing what it would do, and we'd see what was invariant in the network and what varied depending on what the rat was doing. And that seems like a valuable technical goal. And it seems like the goal that has been adopted. It's just that we're not quite to recording all the neurons yet.
What was it in the introduction to the Brain Initiative? Allen and Collins, they wrote, we know that neurons fire, we know that they're connected. We don't know how they act in concert to govern behavior. And then I think there was a fun comment by Andrew Maurer saying that the connectome was absolutely necessary and completely insufficient. And what she was emphasizing, and I guess going to your question, is why? That's all I ask you as a grad student. Why is it completely? Like, do you agree with this statement, and do you think it's completely insufficient? The connectome.
Yeah, I mean, I think the anatomy is one part of it, but then you have the dynamics, which
then you hit on it, right? I think the most interesting stuff that will come out for cell assemblies is the dynamics. So where are the rules that govern the dynamic patterns? Right? And you touched on something awesome. I thought it was like the systems, your level. What level of analysis are you at? Because it's fun to think that neurons make up the brain, but we would never start a meteorological investigation by looking at nitrogen and oxygen, right? We would look at things like vague temperature, which is like a statistical measure of many molecules' velocity, wind. And so you can jump scales quite easily and still be focusing on similar things. But yeah, I think the dynamics is where the most interesting things of the future for. And Jeff John didn't listen when we're live today. I'm sure he'd be infatuated by the dynamic patterns and the sequences that he sees evolving, especially with calcium imaging, which may give way to some insights here.
But you said something about the rules that govern the dynamics. But the thing I was trying to talk about the perfect experimentalist world doesn't tell you anything about the rules. It just tells you what happens.
That's right.
And there's some kind of a leap of faith where we think that if we could see it, if we could see the movie of all the neurons firing at whatever resolution we need, that we would look at that and we would go, oh, I get it.
If we had all the genes and we decoded the entire genome, we would cure every genetic disease.
So I'm wondering I think that if we could watch the movie of the brain doing what it was doing, we would learn a lot of things, but what we wouldn't learn were the rules that govern the dynamics. And so I wonder, is there a more way to query the rules rather than the performance in this situation or in that situation?
I worry that we're so early that the rules are still nebulous. What do you think?
What do you mean exactly by rules, though? Rules governing the dynamics?
So it seems to me that if I knew the rules governing the dynamics, that means I would understand that parameter space that could be. Whereas if I have a movie of an animal doing something and I know what the neurons do at that time, then I know what happens in that very limited situation. It doesn't necessarily allow me to say, well, in some other situation, the movie's going to look like this. But if I understood the rules, then I would be able to predict the movie in any situation.
I have a thought experiment that I always ask graduate students is if I knew ahead of time which neurons are going to fire in your brain when you go to Six Flags across the street, and I know that for a fact, and somehow I hop in my time machine, go back in time, and make those neurons fire in the appropriate pattern, do you have the Six Flags experience? And you've never been to Six Flags in San Antonio. What do you think?
Yeah, I think so.
So you precognate an experience that never happened. Because if you do, then what that says is like every infinite possibility that you could ever experience is already preordained in somehow in the synaptic or the dynamic assemblies of your brain. I think about these things. I'm thinking I don't think that's possible. I don't think I could. I think that there is a finite capacity somewhere that we couldn't pre-program every possibility into it. And so I wonder about these things, and I think like I don't know if we'll ever be able, given the state in one condition, I don't know with what precision we'll ever be able to say like knowing this that the neurons fire like this in this environment. If I take the rat to the next environment, I may have a decent idea. I can make some probabilistic predictions, but no absolute. I think the brain abhors absolute descriptions.
Well, that's right. There's a sort of computability problem that we don't know the answer to that. For that, it's a little bit like the Laplace's clockwork universe. If it really worked like that, we'd be able to predict everything already. So maybe it's a good time for us to stop on that thought of trying to predict what our brain would do at Six Flags and then get the brain to do it.
Let's go to Six Flags.
It's a good. We're not that far actually from here, right?
That's right.
That's a good note worth to
That's
have.
why he used that example. I think. So thanks a lot, Drew.
Yeah, cheers.
And Francesco.
Of
and
course, yeah.
James.
It was fun.
This has been Neuroscientists Talk Shop.